So lovely to be here in person. Makes all the difference, doesn't it? So thanks to the organisers for the invitation. I'm here to report back from CASP15, but I'll explain to you what that is in a second and think what it means for us as crystallographers and structural biologists. So what I'll go through, and I'll go through quite quickly to try to keep to time and pick up any threads in the experiments. I'll explain what CASP is if you haven't come across it before. I'll talk about the CASP15 results, particularly in the tertiary structure prediction category. That's predicting a single chain. That's where we were the assessors this time round. I'll try and give you a flavour of what happened in a couple of the other categories, the quaternary or assembly structure predictions and the RNA categories. But I'll try and give you an idea of where the field is at, which methods are working well, which targets are still difficult, what that means for you, particularly with regard to getting these models and using them for molecular replacement. So CASP stands for Critical Assessment of Structure Prediction. It's an exercise that happens every 2 years since 1994. It's basically been run from the beginning by John Molt, who deserves an enormous amount of credit for everything that's incentivized in protein structure prediction. What happens is experimentalists submit their targets. These are structures they've determined by crystallography or cryo-EM, but before they've appeared in the PDB. And then the organizers send those out to bioinformaticians who try to predict those structures blind. And then there's a group of people, the assessors, who try and objectively look at those anonymized submissions and see which groups are doing well, particularly well in predicting structures. The categories at CASP15 changed quite a bit because at CASP14, you all remember AlphaFold 2 came to an earthquake, really. And so a lot of the categories are changed. They're listed there. The ones in red, I don't have time to talk about. I'm talking mainly about tertiary structure prediction, but I'll also borrow a little bit of material from Ezgi Karaka, who was the assessor of quaternary structure, and Riju Das, who was the assessor in the RNA category. So one of our jobs at CASP15 was to rank groups according to how well they did in structure prediction. So we need a score defined that we're going to use to do that ranking with. We tweaked the existing score slightly. This is the way CASP works. The scores change slightly with time. So we took the CASP14 score and we added in, as you can see, the RELLG, which many of you will know is Randy Reed's coordinate-based method to predict the usefulness of the model with respect to crystallography and molecular replacement. So that's the, is this going to work? Can you see? Yeah, you can just about see the cursors. So there's a bunch of measurements that relate to local and side chain structure. There are others that relate to backbone, and then there are some that relate to global quality, or ASC there is measurement of self-assessment. So assessors, as well as providing coordinates, they provide reliability predictions, confidence metrics, and we assess how well they do that as part of this global score. The details of how we do that are there. It's all based on Z-scores. We don't penalize very bad things because we want to encourage people to experiment. And we also use a sum rather than some kind of average because we want people to try and predict all of them and produce a general method for structure prediction. So the rankings that came out of this are here. There's quite a bit going on in this. These are the best groups are on the left here with the highest sum total of those CASP scores that I just showed you. Red here are what's called server groups in the CASP lingo, so basically automated protocols that run without human intervention. The blue are ones where the humans get involved and do some tweaking. And one of the differences that we saw from previous CASPs is that the automated methods are basically on a par with the human methods, and that's quite different. Previously, methods have always benefited quite a bit from human intervention, but purely automatic methods have now more or less caught up, which is interesting. Most of the methods are based on AlphaFold 2. There's no AlphaFold 3 and there's nothing like it. So the best groups took AlphaFold 2 and used it in particularly clever ways. The top group that wasn't using AlphaFold 2 at all was the Baker group, and of course they were using Rosetta Fold, which is a very similar kind of approach. The top three groups there, just a flavor of how they approached it, all with AlphaFold, all paying attention to MSAs. MSA stands for multiple sequence alignment. So AlphaFold, as you probably know, collects sequences of homologs, looks at the alignment, extracts covariance information from it, and that's important, as you'll see again, for the structure prediction. So they paid attention to these MSAs. They tried different MSAs to try and see which gave them the best models, and that seemed to be key to the very best methods. Two of the top three, interestingly, took AlphaFold predictions of one kind or another and fed them back to previous generation methods. So ITAS has been around in one form or another for quite a few years. The second best group took AlphaFold predictions and fed them to ITAS. Likewise, TR Rosetta X, previous generation, but that seems not to be essential actually, because the very top group was purely AlphaFold based, and all they did different, in addition, as I say, to paying attention to the MSAs, was a little bit of AlphaFold refinement. The green ones here are kind of internal controls. So you probably know ColabFold. That's the implementation you can use through your browser very straightforwardly. There was also a group who ran AlphaFold in a quite vanilla, automatic way and submitted those results. What you can see straight away is that those simple, vanilla, automated AlphaFold runs are not the best available. You can do significantly better than them with more elaborate approaches, although they perform quite well overall. This one down here is a language model method, and I'll talk a bit more about that later on, but you can already see where these three purple bars lie, that they are not yet competitive with the MSA-based methods like AlphaFold. This is an overview of groups along the bottom and targets down the side. Just to pick out one or two things, you can see that we have a block. This is slightly awkward. Now you can see perhaps we have a block of groups who didn't submit for everything, but then we have a huge number of groups who did really well on most of the targets, and then we have a number of stragglers at the right-hand side who were really not competitive. In terms of the targets, again, you can see a lot of red. Red here is good by this overall structure quality metric. So you can see a lot of red. Many targets were well predicted by many groups, but then there's this trickier bunch of targets at the top, which are quite blue, indicating they were only solved well by the very top groups. They were the ones that differentiated the really good groups from the groups that merely did well on most of the easier structures. The color coding here, you can't really see it very well, but AlphaFold was used by most people in one form or another. Just looking a little bit more in detail at this block of really hard targets here, most of them are what's called free modeling. So that's the CASP terminology. Means that for that target, there was nothing similar in the PDB in structure terms, or nothing similar that could be retrieved by sequence comparison. So you might call that de novo or ab initio or template free modeling as well. And you can see that most of these really hard targets were in that category, so there was nothing similar in the PDB. And that's not really a surprise, but it was actually slightly contradicting some of the slightly exuberant conclusions from the last CASP, where people were saying AlphaFold is so good, it doesn't matter if you've got a template or not anymore. And that's not really true. Where you don't have a template, you do often run into problems, and the target turns out to be quite difficult. I'll show you some examples. Also among those harder targets, it was interesting to see there were a lot of viral sequences. That's something that's been seen before. Viruses, obviously the sequences evolve more quickly, and that makes it harder firstly to collect homologs, but also to get a good alignment of those homologs, and that degrades the quality of the information from the MSA. So we spent quite a bit of time with the targets that the method struggled with, because that's kind of where there's room for future improvement. But it's really important, I think, to pause and just boggle at some of the amazing models that were produced. So this is a huge protein, larger than most single, this is a single chain, larger than most single chains in the PDB, mainly quite structural novel, mainly free modeling, and yet this group here produced a model which is just spectacular. I mean, mind-blowing. In terms of progress from the last CASP, this is a sort of key question for us. If you look at the black and the blue lines here, you'll see that this time round, the methods were doing slightly better or at the same level as last time round. That's if you take the best model that anybody produced for any of the targets and you set them out from the easy to the hard and draw a fit between them. So broadly speaking, taking the best model that was achieved by anybody for the targets, performance was comparable or perhaps slightly better than last time. But what was interesting was that there was no single group that performed as well as the best group last time. The best group last time, of course, was DeepMind with AlphaFold, and there was no single group that approached that level this time. And that actually included DeepMind themselves, because although they didn't enter the competition officially, they were invited to submit some informal submissions, and we looked at those, and they were also not quite at the same level as they were last time. So this led us to spend quite a bit of time wondering whether the CASP15 targets were actually a bit more difficult in some ways. One of the ways in which they were clearly difficult, and this is important to know, because if you have a target like this that you want to make a model of and solve molecular placement, you might struggle if it has these characteristics. So one of the clear features which makes a target more likely to be problematic is the NF. So NF, just to explain, is a measure of the depth of the multiple sequence alignment that you can generate for a target. So if you've got loads of homologs in the database, you can get a nice, deep, diverse alignment. You get lots of covariance information, happy days. If you can't, if your sequence is unique or it doesn't have many homologs, then your NF is low and you don't have so much information to extract to help the modeling. So here I'm plotting NF on the x-axis. Targets that are low here are singletons or very only capable of producing a shallow MSA. And you can see that all the targets where even the best groups struggled, this is a measure of overall quality, all of the really difficult targets had low NFs. So that is clearly a complicating factor. A target which has no template and also has a low NF is very likely to be difficult. We also looked at other aspects, and we think there's probably a connection that alpha secondary structure is harder to predict than beta. I mean, not the secondary structure, the actual protein that is rich in alpha secondary structure. And smaller ones we think are perhaps more difficult to predict than larger ones, and you can rationalize both those ideas. And possibly monomers are harder to predict than oligomers, and possibly crystal structures harder to predict than cryo-EM, but we're less clear on that. One example I wanted to show you, especially to this audience, really interesting. You've actually seen this protein before in Gwindaf's presentation. This came as a target to us. This was one that the method struggled with. It's a virus protein, but the reason we think they struggled with this target is because it's a very unusual protein. This was information from the experimentalists. These crystals are incredibly stable, but the protein is really unhappy if you try to dissolve it in solution. And so you can understand some of that behavior, and you look at the crystal structure and you realize it's really very densely packed. The solvent content is only 25%, which is almost as low as it can be. And so what that means, if you think about it, is that the environment of a single chain within the crystal structure is very strongly influenced by that crystal environment, which of course the modeling programs don't know about. So it's actually perhaps not surprising that the modeling struggles to predict this part here, for example, where the structure is very clearly intertwined, interrelated, in contact with other molecules in the crystal. Okay, so yeah, just to sort of clarify that, we think that the slight drop from CASP 14 to 15 in terms of what the best groups can achieve is because there was a small number of really unusual targets which had no template, low NF, and some of these other complications such as low solvent content here. We paid particular attention to language models because these are quite exciting in the bioinformatics field right now. Language models can be used for predicting lots of things, but as you're training them, it turns out that they're implicitly learning concepts of protein structure. So you can also read out a structure prediction for a sequence from a language model. And that appears to be quite exciting because although they're very slow to train, you can predict the structure with a single sequence without that MSA step. So you don't have the CPU or the compute required to generate the MSA. And you also have the idea that maybe you don't need the MSA, a rich MSA, high NF to get a good prediction. So ESM fold and the ESM language model is an example of this. It's known that you can get very high quality models out of this. Like I say, the big advantage is in the speed of these compared to the MSA-based methods like AlphaFold 2. But they also often claim, and I'll show you whether this is true or not in our hands, they also often claim to do well with proteins that are orphans, so singletons where there are not many homologs in the database, and also designed proteins. So where you can't construct a nice high quality MSA, these language models often say, you can come to us and we will produce you a model without that MSA. So we had 3 language models entering the competition. This is an extract from that heat map. So we've got the best MSA model across the top. Again, red is good, blue is bad. So you can see that the 3 language models here, they're not at the same level as the best MSA method overall. Here's ESM, which was the winner by the overall CASP score. And here's UniRes, which seems to do slightly better simply in terms of structure of fold prediction. The ESM method produced the absolute best model by the CASP15 scoring for 4 targets, 3 of which I can show you here. And the interesting little thing to note from that first one is this is a free modeling target. So that means there's nothing similar in the PDB. This is a new fold, and yet this language model predicted it well. And so there was that question, do the language models generalize beyond what they've been trained on? And the answer is yes. The other important question is whether these language models do in fact perform better when you only have a small number of homologs, a low NF. That's what we're looking at here. So here we've got the targets arrayed from a high NF. So we've got lovely, rich, deep multiple sequence alignments over here. And over here, we've got the singletons and the trickier low NF cases. We've got 2 MSA-based methods in blue, and we've got 2 language model methods in red. If the language model methods were working better with these low NF targets, you would expect the red to be doing higher. This is a measure of structural quality. You'd expect the red to be higher on the right-hand side than the blue. In fact, we don't see any of that. And in fact, if you look at the difference between the red and the blue on the left and the right, if anything, it seems to be a bigger gap on the right. So with this set of targets, there really doesn't seem to be any evidence that these language model methods can perform particularly well with low NF targets. And if you just kind of visually track along, you'll see that it seems to be that where the MSA-based methods, the blue ones, struggle, in other words, they drop the quality, then we see the language model methods also drop. So they don't really seem to have any complementarity, any synergy. They can't really fill in any of the gaps the MSA methods struggle with based on this data. So that's a little bit about the targets as a whole. We also said, where you've modeled the structure pretty well, where are those remaining errors and can we explain those errors? So this is an analysis where GDT is over 80, which means the models are pretty good. And we're saying we're measuring error on the y-axis here, and we're asking whether certain regions, certain characteristics produce higher error. One thing we looked at were contacts. So in the orange here, we've got crystal lattice contacts. In the green, we've got chain contacts, and in the blue, we've got other things. And basically the message there is that all of these methods struggle with crystal lattice contacts. So the error, and you see I've put error in quotes there, but the error seems to be higher at the crystal lattice contacts. And I put it in quotes because if you think about it, well, you all know, I think, that in the lattice forms, you can get some force that deforms part of the protein. And so what we're seeing here is a difference between the crystal structure and the prediction. But if the crystal structure has been subject to some force distorting it away from natural structures as the crystal grows, then maybe that's not actually the correct structure. Maybe that's not valid. Maybe this error is not actually an error at all. It's a difference between a correct prediction, perhaps, and something that's been distorted by the crystal growing. Who knows? We also looked at whether errors were correlated with B factors. We saw in the previous presentation that you can sort of infer to some extent that high B factors indicate flexibility. And indeed, when you bin residues according to whether they're in a decile, what decile they're in terms of B factor, the lower B factor deciles have lower errors. So again, that's not surprising, perhaps. We also looked at side chains. The side chain accuracy had improved slightly since last time. We looked at whether you need to predict main chain well in order to predict side chain well. So these are scores for the backbone, x-axis, side chain, y-axis. Low is good. And indeed, if you want to get accurate side chain prediction, you do need to predict the main chain accurately. We also compared the MSA-based methods with a couple of the language model methods. And one of the ESM method struggles a little bit with side chains. The UniRes method struggles a lot. So if you've got a structure prediction from a language model rather than an AlphaFold MSA-based method, the side chains are likely to be lower quality. Of course, the main thing that we do with these, well, main thing that crystallographers do with these models is use them for molecular replacement. It's well established that they're pretty good for that. And again, that's what we see here. So this is all of the targets for which we had the diffraction data and methods, including the best methods, but also ESM as the language model method. You can solve most of those structures with one method or another. You can usually solve them with CASP15. That's the one that you get through your browser or can get through your browser. CASP15 is not the absolute best, but it will often do it. ESM single sequence, this language model method, is not quite as good, but when they're available, and they weren't available for some of these dots are simply the absence of a prediction, but where they were produced, they were often good enough. Often the MR is really straightforward, but sometimes you need to work a bit harder at it, and that's what I'm showing here with some of Ronan's data, that this dotted line here, yeah, you should be able to see that, it is the threshold LLG of 60. And what this is showing is for about half of the predictions, even trimming off the low quality parts, you don't get above that threshold because the models have inaccuracies. This is quite a challenging case where you've got two copies of a multi-domain protein, and essentially the message is that you need to take that protein apart into separate domains in order to solve the molecular replacement. So this is a slightly more challenging case, but it solves with many methods as long as you split the domains apart and treat them individually. There's different ways of doing that, of course, but one program that CCP4 offers is slice and dice. Here it is on that exact case. The ESM method, the protein language, the model overall in global terms is pretty poor. But if you trim off the low confidence stuff, you're left with some domains which are individually well predicted, which you can fit very well. And I've picked out the fact that we use the Birch algorithm to split the domains apart. You're probably familiar with the idea of using the PAE from AlphaFold to do that. That's fine, that works very well. But some of these methods don't give you a PAE, or sometimes the PAE is not so easily accessible. So you need a more general structure-based method to partition the domains. And we have a number of them built into Slice and Dice. The default is Birch, which seems to work quite well. So just to give you a quick flavor, I don't no idea what I'm doing on time here. 5 minutes, oh, right, okay. A quick flavor of two of the other categories. So there's a separate challenge to try and predict quaternary structure. This was the set that people tried to predict. Here, there was an amazing leap from the previous CASP, because of course, in the previous CASP, there was no AlphaFold in this category. AlphaFold was developed to predict a single chain. People didn't know whether it would work well on assemblies. Of course, it does, and the AlphaFold multimer works even better. And so there was a huge leap in success, such that 90% of these cases, you got a decent model, and 50% of the cases, you got a really excellent model. So that's really quite stunning progress. These are slides, like I say, from Esgy. The one interesting thing to pick out here is that that yellow there is the kind of vanilla AlphaFold multimer, and you can, again, do much better than a sort of black box vanilla AlphaFold multimer by working a bit harder, by trying different MSAs, by sampling a lot more models, and so on. In the RNA category, again, there were some really amazing models. This RNA molecule is a lot bigger than it looks, because a nucleotide is 3 times bigger than an amino acid. Interesting to remember, and yet get very good models. For all of the natural RNAs, not the RNA complexes, for all of the natural RNAs, you got something that was the right fold from one group at least. There are 2 different ways in which they're measured, whether it's a right fold. The big, well, slight surprise, I guess, interesting observation was that the top 4 groups there did not use deep learning. So everyone expected that a deep learning method would take over there, just like it had in the protein world. That didn't happen. These top 4 methods here are all more conventional, but the deep learning is only just behind. And in fact, there was a deep learning method mentioned, which is just as good on the natural RNAs as the specific ones. So I think that this is my interpretation of the conversations. I think when the RNA people are talking to the deep learning specialists, there'll be an absolute explosion of progress. So I think that'll be really interesting to watch for next time. Back to proteins, back to MR. How do you find these models if you want to use them for MR? There's a number of ways of doing that. CCP4 provides you with this MR parse, which is quite a nice package. Of course, there are databases of these models. You don't even need to make your own quite often. MR parse will take a sequence. It will give you some information on the diffraction data, perhaps help you spot a pathology, but then it will also find homologs in the PDB, in the AlphaFold database, and it will calculate your model using ESM fold. How to choose between those? Well, it depends on how good the match is in the PDB. If you've got a good match in the PDB, you don't want to forget about that. You have the color coding of the hits in the AlphaFold database and the ESM fold model according to predicted quality. And you try enough until you make it work. Just be aware, and we've seen this from CASP, that the ESM fold models will be less good, and including in terms of their side chain and quality estimates. You might still want to make your own models, because of course the databases only have one isoform. A number of reasons. I will skip over those, you can read them there. So in harder cases, or where you want to access a different conformation that you know to exist, you may want to get more diversity in your models. So this is the last substantial slide. There are different ways of doing that. One of them is very easy. That's to use this Colab page, which I'm sure you're all familiar with. Paste in your sequence, tick a few options that introduce some randomness into the calculation, and you can get back a greater variety of conformations. There are also these other strategies, including ways of manipulating the input MSAs, which will push the AlphaFold in different directions, give you different conformations back. So to conclude, AlphaFold 2 is still the main player, and MSAs are still important. If you've got a singleton protein, you probably won't get a good prediction. Language model methods are not producing as good predictions, certainly not for the hard cases, but they're very quick, and I think that will open up other applications for them. All methods still struggle with some cases, so these are things where there's no template, where there's not many homologs, and these other complications that I talked about. And then another important thing to remember is that if you just run a simple deep AlphaFold 2 or CoLabFold, you'll get a good model, and it may well be a model that's good enough, but it's probably not the best model you can get. So in harder cases where you need to, where you want to invest a bit more time, you can probably get something better by a variety of other strategies illustrated here. The same goes for quaternary structure. Like I say, RNA, I think, is the new frontier. Big things are going to happen there. Yeah, and then like I've just been saying, you can easily link out to the databases, but you may need to work a bit harder in some cases. This is the team that did all of the CASP analysis, including Andre at the CASP organizers and Ronan, who you all know. The rest are in Liverpool, funded by CCP4 or co-funded by CCPM in part, thanks to them. And I'll just finish with this, which is really interesting. This is a poster at CASP. A business school in Spain sent some people to study the bibliometrics of AlphaFold 2, which anyone who's tried to sort of follow all of the developments relating to AlphaFold 2 will sympathize with. This is a whole research area in its own right, just on AlphaFold 2 related stuff, which again is another indication of how important it is to our lives as structural biologists right now. Okay, thanks.